hear me? Right, so I'm going to present the work we did on trying to find a way to find the optimal sparsity criteria for network inference. It's done by myself and Torbjörn Nordling is my collaborator, with the help of Matthew Studham, a postdoc in our group, and our supervisor, Erik Sonnemann. So just to clarify what I mean, so a regulatory network describes the mechanistic interactions of the network, and inferring this network just means finding these interactions that happens between the genes and or the gene product. So this is our network. It's a toy model that we use. It's 10 nodes, 25 links. It's a stable system, and it has up and down regulating interactions shown in blue and red. And this is what we're trying to find. So how do we do this? Well, we do experiments, of course. So we do perturbation experiments in this study, and we apply perturbation and initial condition. We let the system map this initial condition to final state, and we look here at steady state expression. And then from this steady state and the perturbation, we try to work out how the networks actually look. So this is not problem-free. I mean, we both in the perturbation and in the expression, we have noise. I mean, when we measure it, we also have some measurement noise. So there are a number of methods that try to sort of account for the noise. So what happens when we try to work out the system, we might infer links in the network that are not actually there. So these methods try to penalize the inference to try to remove links that are not really in the true final model. So we use this toy model to generate some data, and then we apply here a lasso-based method, and we vary this penalty, this sparsity down here. The numbers on the x-axis is not so important. What is important is that here we have a fully connected network, and here we have an empty network. So if we look at the similarity between our toy model and the inferred network, we can see that in this case, we can actually find an exact similarity between the two models shown here. But this blue curve here is not available to us usually. We don't have the true model to compare with. So in our case, we look at the prediction error, and we can see that the prediction error for the estimate that is closest to the true model is much, much higher than for a much more densely connected network. So our problem is, how do we motivate picking this sparsity over this sparsity? This is the problem we try to solve. So again, noisy data will lead to overfitted models and densely connected network, and many contemporary methods for inferring gene regulatory networks to rely on some sort of sparsity parameter for setting the sparsity. But again, how do we pick this sparsity in the optimal way? So we devised a workflow to try to solve this problem. So there are mainly two parts we contributed with, wrapped around this third part. So the first part here is that we might want to do some resampling of the data. Here we do a leave one out cross-optimization scheme. And we want to know how does each sample affect this when we leave it out, affect the actual prediction error. Then we apply the method of our choice, and we vary the sparsity parameter, and we find all the different models along this regularization path. And for all the models, then after that, we do an unbiased re-estimation of the network parameters. So to go into a little more depth, so if we want to do this leave one out cross-optimization, and let's say this is our expression space or gene space, and we have data that spans G1 and G3 here, and some noise maybe in G2, and we make a model out of that data. The model will also span G1 and G3. So if we then try to predict a sample that has its perturbation or its response in G2, the model has no way of mapping between these two. So we will basically just predict the noise that was inferred in the model. So to overcome this, we measure the linear dependence between the sample we want to leave out and the rest of the data set, and we get a value. And if this value passes our criteria, we have it in our validation set. So here our criteria is just the smallest singular value. So we consider this to be the smallest amount of information we have in our system. So if we now look at an example, so here we use the regular leave one out with all the samples, and here we have our sample selection where we only leave out samples that has passed our criteria, but make the model out of the rest of the data. So it's pretty clear, well, it's maybe not so clear, but that we have gained about 2 orders of magnitude in prediction error in general here. But the main contribution of this step is that we have sort of reduced the influence of noise from samples that the model cannot predict, that we make from the rest of the data. But we have still not moved much closer to the optimal network here. A little bit, but not really significant. So the next step is the unbiased re-estimation. So as you might know, that Lasso, for example, is a biased estimator. So this is the Lasso algorithm. Here we fit the model, or here we infer the model from the data, and then we add a penalty term with the sparsity parameter. We call it zeta here, and we get out a final model with perhaps some parameters set 0. However, when we increase this penalty, more parameters will be set to 0. But not only that, the parameters that are not 0 will be influenced by this penalty as well. So this penalty is a problem for the size of the parameters themselves. So we solve this by inferring the models, but then fixing the structures of the models. So the structures of the models are fixed, and then we do a new least squares fit, but with the structure constant. So we call this a constrained least squares, and we solve this problem for all the models in the regularization path. So now, if we look at an example, we can see that the bias in the model that has increased our prediction error from the true prediction error has disappeared somewhat, and the most likely model that explains the data is now our optimal network. That is, in this case. Right. So what if we have a little bit fewer samples and more noisy data? That is, if we actually have no way of inferring the true structure along the regularization path. So here we have that example. So we have inferred all the models between the full network and the empty network, but nowhere along this path is the true network inferred. So in this case, we have one peak, or actually 2 peaks here. So this is where we use leave one out with sample selection, and this is where we use leave one out with sample selection and constrained least squares. And in this case, of course, again, the optimal network is obscured by this bias in our inference method. But when we then do constrained least squares, our method picks this point. So why does it pick this point over that point? Well, so you can see it this way. If you have a model and you try to remove a parameter in this model that is essential for describing the data, you can kind of predict that the prediction error will increase, and if you don't have this parameter model, you will have a larger prediction error. So this point is actually the most likely model that explains the data. Right, so just to go through the steps again, we looked at each sample if we want to do resampling of the data, so that a model inferred from the rest of the data actually has a possibility to predict the sample that we want to leave out. Then we apply our inference method of choice. We do this leave one out, and then we, for all of the regularization path, for all complexities of models. And then after that, we do an unbiased re-estimation of all the network parameters, constrained least squares, and we find an optimal network structure. So I'd just like to acknowledge my coworkers in our lab, Thomas, Oliver, Gabriel, and Dimitri, and also the organizer for letting me speak here, and thank you all for listening.
So we have time for questions. Anyone?
So I have two questions. One is that have you tried your method on some real data sets like E. coli, fly, or other data sets? And my second question is that when you are putting constraints and you are removing your regularization parameter, so I did not understand what is the constraint. You put a constraint on the topology of the network, or how you basically get rid of this regularization parameter?
So we don't actually create the models themselves. So if you want to use sort of a penalty-based method to get all your models, then you have to trust that that method is actually good in itself. So then we just find the maximum likelihood estimate of the network parameters after the method has been applied.
But the problem is that we don't, in real life, we don't have this, unless you do some cross-validations with some data set to estimate this parameter. But we don't have the true network structure to be able to estimate this maximum likelihood.
Right, so it's a little bit of a problem that we don't have real data. So we haven't actually found any data that can actually, that the first step in this workflow can actually work on. Because usually you have a single gene knockdown or overexpression, and if you leave that one out, that's per definition linearly independent from the rest of the data. So we haven't found any data sets that we can actually use to test our method on. So did that answer your question?
Any other questions? Okay, well, let's thank Andreas again.